Welcome to this lecture on developmental biology. Today, we are going to explore a fascinating topic, the extra-embryonic membranes in birds. Imagine an astronaut floating in the vast, empty vacuum of space. To survive, they rely entirely on a highly complex spacesuit. A bird embryo, developing inside a hard-shelled egg, faces a very similar survival challenge, completely isolated from its mother and the outside world. We will discover how the embryo builds its very own biological spacesuit to survive. Now that we have pictured our embryo as an astronaut, let us look at what this biological spacesuit actually is. These extra-embryonic membranes are living tissues that originate from the embryo's own cells, specifically from the primary germ layers, like the ectoderm, mesoderm, and endoderm. Because they share the exact same DNA, they are truly a part of the organism, yet they are left outside the main body. They function temporarily during the incubation period to manage nutrition, hydration, respiration, and excretion. Just before the chick hatches, these vital membranes are either absorbed into the body or left behind in the discarded shell. Understanding what these membranes are leads us to appreciate their massive historical significance. The evolutionary invention of these membranes was a monumental breakthrough in the history of life on Earth. Before these structures existed, animals like amphibians had to lay their eggs in water to prevent them from drying out and to allow toxic wastes to simply wash away. The development of the extra-embryonic membranes created the amniotic egg. This incredible adaptation allowed reptiles, birds, and eventually mammals to reproduce entirely on dry land, forever changing the trajectory of vertebrate evolution. Beyond their evolutionary history, these membranes have profound importance in our world today. In modern, real-world applications, understanding these membranes is absolutely crucial for the poultry industry. By knowing how this life support system operates, we can optimize egg incubation conditions and maximize hatching success. Furthermore, scientists frequently use the chick embryo's chorioallantoic membrane in medical research. Because it is so rich in blood vessels, it provides a perfect environment to study blood vessel growth and to test new cancer drugs, making it a vital tool in modern medicine. Now that we understand their importance, let us look at the specific jobs of the first two membranes. First, we have the yolk sac, which grows out from the embryo's gut. It surrounds the nutrient-rich yolk and uses a vast network of blood vessels to digest and transport food directly into the embryo's bloodstream. Second, we have the amnion. This is a fluid-filled sac that immediately surrounds the embryo. It acts as a critical shock absorber to prevent physical damage and keeps the delicate developing tissues from drying out during the long incubation period. Moving outward from the amnion, we encounter the final two membranes. The chorion is the outermost membrane that lines the inside of the eggshell, serving as a protective envelope for the entire system. Finally, we have the allantois, a balloon-like sac that grows out of the embryo's hindgut. Its primary job is to store toxic metabolic waste, specifically solid uric acid, so it does not poison the growing bird. As the embryo grows, the allantois expands and fuses with the chorion to form the chorioallantoic membrane, which presses right up against the porous eggshell to handle breathing. Before we move on to how the embryo breathes, let us see this setup with a concrete visual diagram. Here, you are looking at a cross-section of a developing bird egg. Notice the thick outer boundary, which is the protective eggshell. Right in the center is our growing embryo. Below it, you can see the large yellow yolk sac providing nutrients. Immediately surrounding the embryo is the blue fluid-filled amnion, protecting it from shocks. The large pink structure is the allantois, storing waste. Finally, notice the thin chorion lining the very inside of the eggshell, enveloping everything. Having seen the chorioallantoic membrane pressed against the shell, we must ask how it actually functions. Because the bird embryo is sealed inside an egg, it cannot breathe using lungs. Instead, it relies entirely on the physical process of diffusion for gas exchange. Oxygen from the outside air must pass through the microscopic pores of the eggshell and then cross the chorioallantoic membrane to reach the embryo's blood. This life-saving flow of oxygen is not random. It is strictly governed by the fundamental laws of physics, which dictate exactly why these membranes must be structured the way they are. This physical process we just discussed leads us to an important mathematical relationship known as Fick's law of diffusion. This formula explains exactly how fast a gas will move across a biological barrier. Q represents the total rate of oxygen diffusion. D is the diffusion coefficient, a constant based on the membrane material. A is the surface area of the membrane. Delta P is the pressure difference between the fresh outside air and the oxygen-depleted blood. Finally, X is the physical thickness of the barrier. Notice that if the surface area A increases, the oxygen flow Q increases. Conversely, if the thickness X increases, the flow decreases. Now that we have our formula, let us see this with a concrete example of a healthy day 14 chick embryo. We are given a diffusion coefficient of 0.0002. The surface area has grown to 50 square centimeters. The pressure difference is 40 millimeters of mercury, and the tiny thickness is 0.04 centimeters. We substitute these biological measurements into Fick's law. Multiplying the constant by the area gives 0.01. Dividing the pressure by the thickness gives a massive gradient of 1,000. Multiplying these yields our final answer. The membrane successfully pulls 10 cubic centimeters of oxygen into the blood every minute. So we have proven that a healthy membrane works perfectly. This brings us to a critical question. What happens if something goes wrong? Let us consider a pathological scenario during development, perhaps due to a viral infection or a genetic defect. Imagine the exact same day 14 embryo, but due to severe inflammation, the chorioallantoic membrane has swollen. This swelling causes the total thickness of the barrier to double. Because the physical distance the oxygen molecules must travel has increased, we mathematically expect the overall rate of diffusion to decrease, threatening the embryo's survival. Let us apply Fick's law to this new swollen scenario to see the exact impact. All values remain the same, except the thickness in the denominator has doubled to 0.08 centimeters. We set up the equation, isolating the effect of the membrane's thickness. The first part still equals 0.01. However, dividing the pressure difference by the new, larger thickness yields a reduced pressure gradient of only 500. Our final calculation reveals a new rate of 5 cubic centimeters per minute. This is exactly half of the healthy rate, meaning the embryo is now suffocating. We have covered the full topic of extra-embryonic membranes, so let us reinforce the most important ideas. First, remember that the yolk sac, amnion, allantois, and chorion work together as a temporary, highly specialized life support system. They provide food, protection, waste storage, and respiration. Second, as we saw through Fick's law of diffusion, these membranes must grow to cover a massive surface area while remaining microscopically thin to ensure the embryo gets enough oxygen. Finally, never forget that these four structures represent one of the most important evolutionary adaptations, enabling vertebrate life to conquer dry land. Excellent work today!